Hello, my name is Elijah Hernandez, and today I will be presenting a clinical case involving Streptococcus pneumoniae serotype 35B, which is an emerging antibiotic-resistant strain. This case also highlights how newer diagnostic methods, specifically CRISPR-Cas9 serotyping, can be used to accurately identify bacterial strains and guide treatment. The patient in this case was a 28-year-old male who was admitted to Community Regional Medical Center in early January after experiencing about 1 week of fever and cough. His medical history included a thyroidectomy due to hyperthyroidism, but he was not considered immunocompromised. Another relevant detail is that the patient frequently went on late night runs in cold weather and may have contributed to respiratory stress. He also previously received a pneumococcal vaccine, which becomes important later when discussing strain resistance. Upon physical examination, the patient presented with was presented with several severe symptoms. He had a high fever of 104 degrees Fahrenheit, a strong cough, and was producing brown sputum, which suggests a bacterial infection in the lungs. His oxygen saturation was measured at 94 degrees. I'm 90. No. His oxygen saturation was measured at 94%, indicating mild respiratory compromise. In addition, he had a chronic middle ear infection, which caused redness and muffled hearing due to fluid buildup. Uh, Altogether, these symptoms pointed towards a serious respiratory infection with possible complications. Based on these symptoms, clinicians initially suspected bacterial pneumonia, most likely caused by Streptococcus pneumoniae. This patient was immediately started on penicillin, which is a standard first-line treatment. However, the patient did not show improvement, which raised concern. Because of the severity and persistence of symptoms, there was concern that the infection may have spread to the cerebral spinal fluid, potentially leading to meningitis. This prompted further diagnostic testing. Several laboratory tests were performed to identify the pathogen. The patient's blood work showed an elevated white blood cell count, indicating an active infection. Sputum samples were cultured and showed alpha hemolytic growth on blood agar, which is a characteristic of Streptococcus pneumoniae. The organism tested negative for catalase and did not grow on mannitol salt agar nor McConkey agar, helping rule out other bacteria. Additionally, imaging showed dark spots in the lungs, indicating bacterial colonization from the x-rays. Altogether, these findings strongly supported a diagnosis of streptococcal infection. To, to precisely identify the strain, CRISPR-Cas9 serotyping was used. This method targets a specific region of bacterial DNA known as capsular polysaccharide, or the CPS locus. In this case, the CRISPR system recognized the region between the DexB and ALI-A genes, which is about 20 kilobases long. The Cas9 enzyme cuts this region, and the DNA is then sequenced using nanopore technology. The sequence is reconstructed and compared to known databases to determine the exact serotype. Using this method, the pathogen was identified as Streptococcus pneumoniae serotype 35B. Streptococcus pneumoniae is a gram-positive bacterium that normally resides in the nasopharynx. However, it can become pathogenic and cause disease such as pneumonia, ear infections, and meningitis. The strain identified in this case, serotype 35B, is particularly concerning because it is multi-drug resistant and is becoming more common due to changes driven by vaccine pressures. One of the key issues in this case is antibiotic resistance. This strain shows resistance partly due to capsular polysaccharide replacement, which occurs when vaccines target certain serotypes, allowing others to emerge. Additionally, the bacterium has altered penicillin binding proteins, which reduces the effectiveness of beta lactam antibiotics like penicillin. This explains why the initial treatment failed and why stronger combination antibiotic therapy was needed. Streptococcus pneumoniae also has several variance factors that allow it to inflict the host. First, the capsular polysaccharide protects the bacterium from the immune system and harsh conditions. Second, IgA protease breaks down IgA antibodies in the respiratory tract, which allowing the bacteria to evade immune defenses. And third, pneumonolysin is a toxin that forms pores in host cells, causing cell damage and death. These virulence factors work together to allow the bacterium to colonize the lungs and cause severe infection. Due to antibiotic resistance, treatment had to be escalated. The patient was given increased penicillin along with cetra, uh, cetraxone, um, moxifloxacin, acin, and vancomycin. After this combination therapy, the patient showed significant improvement within 3 days. He was discharged after 1 week and fully recovered within about 3 weeks. This demonstrates the importance of adjusting treatment when resistance is present. This clinical case highlights an important issue in modern microbiology. Pneumococcal vaccines, while effective, can lead to capsular replacement, allowing new serotypes like 35B to emerge. This contributes to increasing antibiotic resistance and makes infections more difficult to treat. It also emphasizes the importance of advanced diagnostic tools like CRISPR for identifying pathogens quickly and accurately. And in conclusion, this case demonstrates the growing challenge of antibiotic resistance, Streptococcus pneumoniae. Accurate diagnosis, understanding of virulence factors, and appropriate treatment strategies are critical for managing these infections. As resistance continues to rise, both diagnostic methods and treatment approaches must continue to evolve. And here are my references, and that concludes my presentation. Thank you.